Institute for Mathematical Sciences. Thanks for joining in this plenary talk, which is a webinar. It's live streamed. So my role as the director is to make sure activity happens in the Institute. And as you know, during lockdown, our normal activity can't take place. So it's fantastic that we've been able to pull together such a brilliant virtual program on IDP, on the infectious dynamics of pandemics, and this is just one small talk as part of that 4-month program. Of course, life will continue eventually back to normal, and so I would urge you in the audience to look at the program of activities in the future when hopefully we can restart physical meetings, and there's plenty more to come through till 2023. But also I'd like to put out a plea for all of you while you're in lockdown to think about other things that the Newton Institute can and should do at this time, and we're always open to good ideas, or we're open to criticisms if you think there's things that we're not doing very well. But if you also think there's programs that we could be doing in the future which may be related to epidemic modeling or otherwise, then just get in touch with me. We're very happy to help. And just finally, I should say that thanks to the wonders of persuasion, mathematics is getting some more money. So in coming years, we hope that the Institute will have some more monies to be more adventurous about what we do in our programming. So again, I'd urge you to take advantage of that. With that, I'd like to thank all the organisers of all the events so far, and particularly to thank Robin, who's going to be our host, master of ceremonies today, and Adam for giving the plenary talk. So thanks, guys.
Thanks very much, David. Yeah, it's great to be able to introduce Dr. Adam Kucharski, who is giving today's plenary talk. He is an associate professor in infectious disease outbreak modelling at the London School of Hygiene and Tropical Medicine. He did an undergraduate degree in maths at Warwick, after which he went on to Cambridge to do a PhD in mathematical epidemiology. After that, he went on to a postdoc at Imperial, and he's been at the London School since 2013, first with a fellowship from MRC, and then now with a Sir Henry Dale Fellowship funded by the Wellcome Trust and the Royal Society. He has a range of other related interests. So, for example, he's an excellent author as well. He's been working on COVID-19 since the very start of the pandemic. And I think it's fair to say that he's one of the voices of mathematical modelers during the pandemic. So in particular, I checked it out beforehand, and he has just under 88,000 Twitter followers, of which I am one. So, so thanks very much on behalf of me and all of your other followers for a fantastic comments via Twitter throughout the pandemic. The only other thing to say is that for workshop participants, please submit questions for Adam, which he'll take at the end via Slido. And with that, thanks very much to the Newton Institute, and thanks also to Adam again for giving this talk. So I'll hand over to Adam now. Thanks.
Right. Well, thanks for inviting me to give this talk. I want to really dive a bit more into how we measure disease transmission. I think reproduction numbers are a concept that perhaps 6 months ago were not so widely known outside of our community. I think it's a concept that's really far more prominent now. So I want to look a bit at the history and some of the nuances of actually how we go about calculating it, and what some of the implications might be for control, well, now and over the coming months. So first, a little bit of history. You know, when we're talking about measuring transmission, where do these ideas come from? And a lot of them can be traced really to 100 years ago, and a lot of the early work on disease transmission and modeling was around malaria. And one of the pioneers of this, Ronald Ross, who constructed a lot of the early models, after initially making this link that mosquitoes transmitted malaria, wanted to understand control measures. And so he would develop these conceptual models of transmission. And in his book in 1911, suggested this hypothetical situation. So he wanted to understand the effect that mosquitoes might have on malaria outbreaks, and said, let's suppose we've got a village with 1,000 people, one of whom has malaria. And in his sort of conceptual model, he assumed that for simplicity, let's say that there were 48,000 mosquitoes in this village. And over a given period of time, perhaps not every one of these mosquitoes will bite someone. He assumed, well, perhaps 1 in 4 of these mosquitoes might bite another person. You've got 12,000 mosquitoes that bit someone. But of course, there's only 1 in 1,000 people in this village actually has malaria. So on average, we'd only expect 12 of these mosquitoes to actually bite someone who's infected with the disease. And then it takes some time in the mosquito for the parasite to become infectious, and not all mosquitoes will survive that long. So Ross assumed that only 1 in 3 would actually survive long enough to become infectious. So you've got 4 infectious mosquitoes, and then of course, as I mentioned at the start, we're assuming in a period of time only 1 in 4 mosquitoes will bite another human. So you end up in a situation where you've got 48,000 mosquitoes, but only 1 of them actually end up biting the infected human and going on to bite someone else. A lot of the early work in modeling was really around these kind of focus on densities, a lot of work on malaria, a lot of focus, even as it expanded out into other diseases, the idea of sort of population densities and how much do you need for transmission to persist? Because of course here, you've got 1 initial person in this village, and then through this whole process, you've only got 1 additional case. So if that initial person recovers or is no longer infectious for some reason, your outbreak isn't really growing. But it would take some time for people to really generalize these ideas and apply them to other diseases. And a lot of the progress came in the 1950s. Another researcher working on malaria, George MacDonald, built on some of Ross's modeling approaches. In particular, at the time, WHO were launching a lot of interest in eradications. The idea was, could they actually wipe malaria off the face of the planet? And MacDonald's models, again, focused on these different properties of the mosquito, of how the parasite spread between those and humans. But in the appendix of one of his papers, he came up with this suggestion of a concept. He called it the basic reproduction rate. And it was almost a kind of offhand suggestion at the bottom to think of maybe thinking that the number of new infections that come from the presence of a single case. So in other words, if you have an infectious person, how much transmission is being generated. But there's an idea that didn't really take off that quickly. And it really wouldn't be for another 20 years or so before it really rose to prominence. And a lot of it was the ability to apply these ideas to data, to other diseases, particularly with the emergence of things like HIV, the need to be able to quantify transmission. When we talk about something being transmissible, being able to put numbers and actually measure that. And so this idea would gradually evolve. Its name would also change. I mean, although MacDonald called it the reproduction rate, it actually doesn't depend on time because it's just the number of infections. So often in our field, we would now call this the reproduction number. And as I'm sure is familiar to a lot of people now, this is just defined as the average number of secondary cases that originates from a typical infectious individual. And the reason this is very useful is, rather than historically where people would talk about mosquito thresholds and perhaps quite abstract notions of whether a disease is under control or not, the reproduction number gives us a really clean threshold for whether we expect the epidemic to grow or not. Because if on average each case is generating more than 1 additional case, if R is above 1, we're going to expect to see growth over time. If it's equal to 1, we'd expect the epidemic to stay flat, because like that village example I showed, each case is just generating 1 new case. And then if the reproduction numbers below 1, we would expect to see some decline over time. And obviously the value of R will give us an indication of how much on average we expect that growth or decline to happen. But then it brings us to the question of how do we actually estimate this number? We're still in quite theoretical, abstract territory. Well, if we could somehow observe every single infection and transmission event occurring, then the task would be quite simple. If we were in a world where somehow we could literally see every single transmission event and work out how all these infections are linked, we could just count up the infections per person, work out the average, and then we'd be done. But unfortunately for biological infections, certainly we can't get this level of insight generally. It's very difficult to really see each one of these transmission events. So we have to find some ways, instead of estimating reproduction numbers, indirectly from available data. And one of the ways it's very common to do that is to look at the timescale of growth of the epidemic and then piece together that timescale with the magnitude of growth to give us an estimate of what this reproduction number might be. So it rests on this basic idea that if you have a case and they infect some other cases, there'll be a timescale on which that process occurs. And one way to capture that is by what we would call, for example, the serial interval. So you might have a case appearing with symptoms, they infect some individuals, and then those individuals appear with some symptoms. And then that gives you a timescale of the growth of your epidemic in this very simple initial situation. So over that period of time, you might expect the growth to be generated by this R value, that if one individual, for example, gives it to 3 over that timescale, the level of growth you see will be a measure of the reproduction number. So to give a very kind of simple illustration based on this logic, if a serial interval is say 1 day and an R is 2, then you might expect just a very simple doubling. So on the first day, perhaps you get 2 new people showing symptoms. And then after 2 days, you get another 2 people for each of those additional cases. You get 4 new cases, and then you get 8 growing over time. So in other words, after 3 days, under this very simple step-by-step model, you might expect 8 times more cases than you started with. And this kind of logic is very useful because often we don't know what the reproduction number is. Here, I've just made up an assumption. I've assumed it's 2 just to kind of show you what this looks like. But for many infections, particularly things like COVID that emerge and we don't know how transmissible they are, we have to try and estimate R. So we can use this exact same logic to kind of work backwards. If we know what the growth has been over time, if we can estimate what the serial interval is, then we can get an estimate of what we think the reproduction number might be. So the basic logic is as follows: that if we have some idea of when the outbreak started and how many cases there were, and if we have some idea of how long this serial interval is, then if we know eventually how many cases there are after a given period of time, by looking at this exponentially growing case curve and the timescale it's occurring on based on a serial interval, we can get a rough idea of what the reproduction number might be. And this is the sort of approach that a number of groups used early in the COVID outbreak to try and work out what R was. So this was back in mid-late January. But the problem is, in particular, this part of the equation. How many cases have there been over time? It's something that certainly in the early stages of the COVID outbreak was difficult to answer, because a lot of cases, it seemed, weren't being detected and hence weren't showing up in data. So back on the 16th of January, there were 41 confirmed cases in Wuhan, but a report by Imperial noted that already 3 cases have been detected abroad. So even just from a purely sort of probability point of view, if you only have 41 local cases in outbreak, the chances that 3 have already been exported to other countries is pretty unlikely. And what they did is they put together a fairly simple probabilistic model based on flight connections coming out of Wuhan, and worked out, well, if we go backwards, if there's 3 exported cases already, how many cases are there likely to be in Wuhan to have already generated that number of observed exported cases? And the analysis had so obviously some uncertainty around it, because 3 cases is working with quite small numbers, but suggested that there were already probably thousands of cases in Wuhan by that point. And indeed, over the coming days, the number of reported cases gradually increased. And this gave people the data they need to use that kind of approach that I showed, where you can look at that growth. If you've got an estimate of when the outbreak might have started, if you've got this endpoint where it's got to so many thousands of cases, and you can make some assumptions about the serial interval. A lot of these early studies were using, for example, values from things like SARS, which were related viruses, then it's possible to get some rough estimate of what transmissibility might be. It's worth noting that it's not just the early exported cases from Wuhan that revealed the size of the outbreak. For example, exported cases from Iran, US, even UK were reflecting that perhaps domestic outbreaks were larger than the raw case numbers suggest. So throughout this outbreak, we've seen, particularly in the early growth, case numbers not necessarily reflecting the underlying number of infections. So going back to the Wuhan outbreak size, a number of studies tried to estimate what R was. I thought this was quite a nice illustration in one of just the uncertainty that's involved, because as I said, you need to know when it starts, the initial seeds and the initial number of cases, and how big it is eventually, and of course what the serial interval might be. And all of those things are uncertain at the start of this outbreak. So this study reflected that uncertainty in the modeling, which is why you've got all these different potential trajectories. But across this uncertainty, they estimated reproduction number potentially around 2, but could be anywhere between about 1.5 to around 4. So quite a large range of values, but subsequent more refined estimates generally seem to sit in this range, probably more towards the sort of 2-3 value, depending on where transmission is occurring. So this is a very useful early estimate in that initial growth phase of the outbreak, what R might be. But of course, we've seen R change considerably over time for this outbreak. It's not some inherent fixed value that it's always going to have this within a population. Of course, it may well be above 1 initially. You might see this exponential growth as we saw in a number of countries early on. But policies and control measures will change that. In a number of areas of the world, control measures have brought the reproduction number below 1. And then, of course, it may be that countries ease measures and subsequently see a change in transmission. And depending on how much those measures are eased and how much behavior is changing, it may be that the reproduction number ends up increasing quite substantially. It may increase slightly. It may increase only a bit and you end up with quite a flat outbreak, or you might end up with a decline. So obviously this is a complete illustrative figure, but just really shows that we can't think of this as a fixed value, that we have to have some way of estimating how it's going to be changing over time. And we can use the same logic that I showed before, that if we have an idea of what the timescale is over which infections are occurring, we can look at the ratio of growth over that timescale to give us a sense of what the R value is at a given point in time. But this is where a few nuances come into play, which I think are quite important to understand if we're interpreting how these R calculations work. And one is, as I mentioned, the serial interval. So we define this based on symptoms because it's something that's nice and easy to measure. If someone's got symptoms appearing and someone else has got symptoms appearing, that's something clinically you can work out the timing of. But in observed studies of the serial interval for COVID, sometimes you see evidence that it can be negative, which perhaps at first glance doesn't really make sense why you would have sort of seemingly one person appearing before the person or after the person that they've infected. And this is why I think we need to distinguish between two related concepts. So one is the serial interval and one's the generation time. So serial interval, as I said, is the gap between symptoms appearing, and the generation time is the gap between the time of infection of the first case and the time of infection of the second case. So just to talk through this, as I said, some negative values, which I'll explain in a moment. Look at the timeline. We've got the first case, in this case, infected. Let's say that after a given period of time, they infect someone else, and then that first case becomes symptomatic. So we know for coronavirus that people can transmit either when they've got very mild symptoms or before they show clear symptoms. This is a bit of a simple illustration, just as a timeline, so don't read too much into the exact values, but it's to illustrate how you can get this feature with serial intervals. So it might be that after that second person becomes infected, for some reason they develop symptoms a bit quicker. So first of all, we've got here the generation time is obviously from that first infection to the second, but the serial interval might actually, in this case, be negative because the first case becomes symptomatic after the person they've infected. So generation time always has to be positive because obviously if you're infecting someone else, those events have to occur in a sequence. But depending on how people are infectious relative to what we'd call the incubation period, so the time between infection and becoming symptomatic, you can have a situation where the serial interval isn't necessarily the same as the generation time. And this is quite important because if we're talking about infection and transmission, it's really these generations of infection that we're interested in. So we need to kind of think about how we can adjust from individuals showing symptoms to actually when the infections are occurring. So if we're talking about transmission, that's really the events that we're interested in. So there's two main methods that people use to try and calculate how reproduction numbers change over time, or particularly how infections change. And so one is this method where you assume that there's an individual that's becomes infected today. So this is TI, it's time of infection of this new individual or group of new individuals. And you kind of look back in the past and you say, well, of all these infections that occurred in the past, each of those might have some probability of infecting the person who's infected today. So if someone shows up infected today, perhaps they're infected by someone a few days ago or longer ago, and there's some probability distribution telling you when that might have happened. And then that person may later show up with a symptom onset. So TS reflects that there may be this delay to show symptoms. The other way we could do it is to kind of look forward in time instead. So there's two illustrations here that kind of have a similar logic. So we could, in B, we could say, well, individuals today who are infected will infect others in future based on some distribution. So if you're infected today, you might be quite likely to infect someone in the next few days, maybe less likely to infect them further in the future because you're getting better and you're less infectious, for example. And then in the final plot C, we might do something similar, but we might do it based on symptoms instead. So we might say someone becomes symptomatic today, and then if it's a disease where transmission happens often post-symptom, then by the same logic, then others will show symptoms at some given period in time. And this is quite a subtle distinction, but quite an important one, because if you think of what's happening in A, we're using past data to try and understand what's happening today. So we've got infections occurring today, and we're using a dependency on infections that happened in the past to calculate a reproduction number. In B, we're looking forward. So we're looking at infections today and then looking at who they infect to calculate what transmission might be. And this is, I think, an important distinction, because when we say, for example, the reproduction number today is 2, what do we mean? Do we mean that individuals who were infected today were the result of a reproduction number that was 2? So you have 2 new cases today because the reproduction number is 2 that generated those cases. So that would be the kind of the method in A. Or are we talking about people who are infected today, they're going to go on in future to infect 2 other people? So that's in B. And again, it might seem like this is quite a fussy detail, but particularly when we're talking about control measures and, you know, you get a change in transmission, what control measures worked, making this distinction about are we looking forward or backward in time, and actually what do we mean by this is quite important. A lot of the measures that, a lot of the methods that are used for COVID, we use this method A, and the reason is that it doesn't require data from the future to make that calculation. That you can actually look at TI and the infections that occur, and you can look at infections that led to that, which means that you're able to make more use of your data as it is. And this is what a lot of studies do. This is some work by colleagues of mine, other groups that are using similar methods. And the first thing they're doing, if you look at Singapore, for example, is they're taking the reported cases and then they're estimating based on the incubation period, based on the delay to cases showing up and based on the time for people to become symptomatic when those infections originally occurred. So that's the kind of blue line here. You're seeing that it's the shifting essentially the gray reported cases to when it's likely those individuals were infected, and then using that infection curve to estimate how R is changing over time. So you can see, for example, in Singapore, early on through February, R was probably around 1, very low numbers of cases. You got this very clear increase in this period where you got this cluster occurring, in this case, in a lot of migrant workers, and then a gradual decline over time. Iran is, again, another example where you had an initial increase in transmission, the reproduction numbers come down, and then increased again. But of course, all of these methods are very dependent on the data you have available. And during this period where a lot of transmission was occurring, certainly within Europe, the level of cases that were being reported was quite low. Now, as long as the proportion of cases that are showing up in your data is sort of consistent over time, so yeah, even if you're missing a lot of cases, as long as you're consistently missing them, then because the reproduction number is really just looking at this growth or decline over time, you'll still get a sensible number. But obviously the concern is that if a country suddenly improves their testing and suddenly starts detecting a lot more cases, then your reported cases might increase, but actually the underlying infections aren't. And so that's why people are really looking at a number of different data sources, all of which kind of affect this estimation in different ways. So for example, this is a timeline of values that you might measure if you're trying to look at transmission. So you've got the infection event that occurs first, and then you might have, after a period of time, someone shows symptoms, they might go to a GP or get outpatient tested, admitted to hospital, perhaps to ICU, and then you might have death or perhaps recovery later on in time. And typically events like hospitalizations and deaths will be perhaps a more consistent measure of the level of impact something is having, because it's not necessarily so dependent on a sudden rise in community testing, for example. But the problem is that those events from an estimation point of view occur much further in the future. So if you're wanting to use data on hospitalizations or deaths to estimate transmission, you're using data that reflects events that occurred far in the past. Infections perhaps even occurred 3-4 weeks earlier. And what this can do, because there's obviously uncertainty in the time delays involved in this, it can have the effect of smoothing your data. This is from a nice recent review paper showing that if you've got the true value of RT, which is the black line, so this is R changing over time, and then you try and use cases that occurred later or deaths that occurred later and try and estimate back from those what the reproduction number is, you can see that the shifted death data can lead to this smoothing effect, because you've got this uncertainty in the delay from infections to deaths, and then you've also got this uncertainty in the reproduction number. So it shows that there is this trade-off that depending on which data set you're using, the delays that are involved, that can introduce uncertainty in your estimates. And there's a lot of work that's going on at the moment trying to find the best combinations, especially as we get to low case numbers, to get a reliable signal, but also a quick one. And that's really a crucial point, having something that's prompt, because in real time, we don't want to wait weeks to find out what the effect of transmission is. If you're waiting 3-4 weeks to find out if your transmission's increased, that's an incredibly late and frankly bad metric to be working off. So there's similarly research effort to try and get this in real time. And I mean, first thing that we perhaps should look at first, though, is what influences the reproduction number. So as I've said, we're trying to estimate it faster. Are there components of it that can provide clues to be able to do that? And what we can do is break it down into, there's a number of different expressions, but for simplicity, I often call these the dots, because I think they're a bit easier to remember. And you can think of what influences are by essentially the different steps that happen when someone becomes infectious. So for example, someone becomes infectious with a pathogen. What influences secondary transmission? What influences the number of new cases? Well, one is duration. How long are they infectious for? The opportunities for exposing others during that period of infectiousness. But also during each of those opportunities, whether it's a conversation or interaction, what's the probability that transmission actually occurs? And then finally, what's the susceptibility of the other person? So you could have an interaction where someone's infectious, they cough, virus gets across, but the other person may or may not be susceptible. So it's 4 things: the duration, the opportunities while infectious, the transmission probability, and then the susceptibility. And if we multiply those average values together, we get an estimate of the reproduction number. And this is useful, as I mentioned, in real time, because some components of this we can actually measure. So in particular, opportunities, if they're reflected in social interactions, which is plausible for a lot of respiratory infections, that's something we can go out and ask people. We can see how much that's changing over time. And that's what some colleagues at London School have been doing over the last couple of months, really, is look at how opportunities for transmission are changing. And because looking at this formula, these things just scale off against each other, that if opportunities halve, then if you kind of adjust sensibly for how those opportunities are occurring within a population, you can work out how much your R might have come down as well. So one study that was conducted immediately in the days after lockdown came in the UK compared 2 values. So on the right-hand plot here, the polymod plot, this is what you could think of as typical social contact. So how many people do you have conversations or physical contacts with, if you're a given age, with other age groups. You can see young groups tend to have lots of contact with young groups, fewer contacts in old groups. It's less likely that young people will meet a lot of old people. And on the left-hand side is what's known as the COMIX data, which is the post-lockdown study. So even just looking at the plots, you can see that there's a lot less intensity of interaction, and actually it's about a 74% reduction in contacts over this period of time. It's not as much as was seen in other areas. For example, one study in Wuhan after their lockdown found that there was about an 87% decline, but still 74% reduction in contacts is substantial. And when that's compared to a sort of plausible estimate of the baseline value of the reproduction number, and when you sort of scale down contacts accordingly, this early study found that if you, depending on which data set you use, but if you use all contacts, including conversations, which is the darker distribution here, the model was fairly confident that the reproduction number was probably below 1. Now, bear in mind, this was a period of time where everything was still increasing, even in many areas of Europe, but really provided an early signal based on social contacts that the reproduction number was likely to be below 1. And indeed, over the coming weeks through March and April, we did see that decline manifest in the other data sets. And work's also been done on various other data sources to try and show these kinds of effects. For example, in Citymapper data, this is from Paris, from Google Mobility, really seeing this drop off in social contacts. But the challenge we have now, and it was there was a nice paper by Kylie Ainsley and some others from Imperial making this point, is that as countries move back and try and relax measures in a way that doesn't lead to new flare-ups, you would hope that there would be a decoupling between these kind of data sources and between transmission. So early on, everyone reduced their contacts and we had this big reduction in transmission. But as countries go back to normal, you could almost view a successful lifting of lockdown as one where all of our mobility and activities go back to normal, but we don't see an increase in transmission. So I think it's a really useful data source initially, but we do have to be a bit cautious now that if there's an increase in Google mobility, it might not necessarily correlate with transmission, because obviously our behavior and the structure of our interactions are going to be very different now. But as well as giving us kind of insights into this early drop in transmission, this framework of separating out the components of R can also give clues about how we might control an infection. So you've got 4 components, and these are all things we could target with different measures. So for example, to take something like HIV, you could, if you get people on effective treatment, you can reduce the viral load, you can reduce the effective duration of infectiousness. If people have fewer partners or fewer concurrent relationships, that can reduce opportunities. If people practice safe sex, that can reduce transmission probability. And obviously recently there's been an increase in things like pre-exposure prophylaxis that can reduce susceptibility. So each of those components in turn will have an effect on the reproduction number, and often in control programs, multiple methods are used together, because it might be that there's not a single measure that can get the reproduction number below 1, but because these things multiply together, a combined approach can help you achieve control. It's likely we'll see similar needs for combined approaches for COVID. So for example, if people self-isolate when ill, effectively it reduces their duration of infectiousness. You know, they'll still be infectious, but because they're not interacting with others, effectively that reduces how long they can transmit for. Social distancing in general reduces our opportunities. So even though people might be infectious, they're just meeting fewer people when they're out and about. Things like face masks and certainly PPE may reduce the transmission probability by some amount. It's not clear exactly how much, but there's various evidence and data sources pointing towards at least some modest reduction potentially from this. And then the ultimate goal would be to reduce the reproduction number by reducing susceptibility through a vaccine. Because if we can get the reproduction number below 1 by reducing S, we don't have to worry about this other stuff. We don't have to interfere in people's behavior. And that's what we do for lots of other diseases. We generate immunity in a population, and we don't have to have people making huge disruptive changes to their lives. And that focus on vaccine is why we often in our field refer to another value to try and study that property in more detail. So as well as reproduction numbers R, we also talk about the basic reproduction number, or R0. And basically that reflects the first three terms in this equation. And what it allows us to do is just summarize everything in the equation that isn't susceptibility. And then we can very easily study what we need to get the disease under control. So if we sort of bunch that all together in terms of R0, we get this R0 times S is the reproduction number. And then obviously to control a disease, we need R to be below 1. So a quick little bit of maths, we can kind of just rearrange this and say, well, if we want, I've just realized there's a typo there, but look at this formula at the bottom, and then I'll flip to the next one. If R is below 1, then we need S to be below 1 over R0. So it's just this formula at the bottom, and we just flip the terms around and say, well, if R is below 1, that's how small we need to get S to make that work. And so in other words, if S is below 1 minus R, then if the proportion immune is greater than 1 minus this value, because immunes are the people who are not susceptible, this gives us what's known as the herd immunity threshold. So if R0 in this case is 2, so if everyone's susceptible, each case is infecting a couple of others, in this very simple average framework, the threshold would be 50%. And there's obviously been a lot of speculation about whether this value might be slightly lower depending on population structure. There is a lot of nice modeling exploring the different properties of this. And that's something I really want to talk about a little bit more, because here we are focusing very much on averages. We're talking about average amount of transmission. We're talking about average interactions. And that's not how diseases obviously behave. You know, we see substantial individual level variation for a lot of infections. If you look, for example, at transmission change in Hong Kong, it's a really nice study that came out not too long ago. You see that there's certain events that have generated a lot of secondary cases. So each circle here is a case, and each arrow shows a transmission event. So you've got these 3 clusters in a bar, in a wedding at a temple, where a lot of transmission occurring. And then you've got these other much smaller clusters also occurring. And there's 2 ways we can measure the extent of, or 2 main ways, the extent of individual variation in transmission. The first is we can just look at this first step. So we can work out these index cases, and then at that individual level, just how many cases do they generate? And that's what this Hong Kong study did, and just said, well, if you've got a case, how many secondary cases are they generating? And just fit a distribution to data. So often people use things like negative binomials to capture that variation at the individual level. And this study found that about 80% of transmission comes from about 20% of infections. If you can't get that very fine scale transmission chain data, another method you can use is to look at the outbreak size. Because if you have a lot of individual level variation, the distribution of outbreak sizes you see will be different, because you'll see a lot of quite small outbreaks, and then occasionally you'll see a larger one. That provides information about the variation in that transmission process. So that's work that some folks at Johns Hopkins worked on, and we also built on some of the things that they were doing, and estimated based on these outbreak sizes of these early infections that about 80% of transmission probably coming from around 10 to 20% of infectious diseases, infectious individuals. And there's now quite a lot of studies that put this in this kind of range. About 80% of transmission, particularly in these early stages of outbreak, seems to be coming from somewhere in the region of 10 to 20% of infections. But then there's the question of how much of that variation is coming from an individual, is coming from a behavior, an environment. And that's really where we need to think more about where are these clusters happening and how are they happening. There's some work from some colleagues of mine looking, for example, at transmission clusters. So where you see these outbreaks, are there certain environments that are showing up more often in the data? And it seems very clearly, certainly in reports, that indoor environments are showing up a lot more. We see a lot of reports of things like food processing plants. There's obviously been a lot of transmission in shared accommodation, in care homes, particularly around close-knit social gatherings, so things like religious events. But of course, there are going to be additional clusters that aren't reported that we need to sort of delve into in more detail. But I think really identifying the features that can generate more transmission is going to be incredibly important if we want to try and target control measures. Because if you go back to this expression for the components influencing R, obviously if R has a lot of variation, by definition, that means that one or more of these components of R must have a lot of variation. So is it the case that there's just a lot of variation opportunities, that some people just have a lot more contacts than others? Or is it that in the setting those contacts are made, the transmission probability is very different? So, you know, someone who's infectious in a gym might have a very different risk to others as someone who's infectious in a park. Or is there some variation in terms of the duration of infectiousness, or perhaps even in terms of susceptibility as well? So there's obviously a lot of work going on at the moment to try and work out which of these factors are important, and crucially how predictable they are, because ideally we want things we can act upon. If we can identify features of transmission we can do something about, that gives us a more targeted option for control. But just to go back to this picture, it highlights, I think, also a paradox with these kind of super spreading events, because you can see that when these super spreading events occur, you get very rapid transmission. But a lot of individuals don't spread much infection to others. And actually, if you have this really dispersed distribution, a large proportion of individuals aren't going to be generating any secondary cases, and a small proportion are going to be generating a lot. And what this means is that if you have a new outbreak, having that variation in transmission actually makes it less likely that a single case will lead to sustained transmission and cause an outbreak. Because if you have a situation where only a small fraction of cases are generating most of the transmission, then if you introduce a single case into a new population, perhaps an imported case, unless a super spreading event occurs, it's unlikely you'll get sustained transmission, which means that a lot of the imported cases won't go any further. And so in a paper we did quite early in this outbreak, we showed that if you have super spreading to the extent of something like SARS or MERS, because at the time we didn't have a lot of data for the new virus, you can see that actually it's less likely that you'll lead to a large outbreak with a single case than if you have something where transmission is much more homogeneous and each case is much more likely to generate transmission as others would. And based on plausible values of the early reproduction number, based on SARS-like super spreading, we estimated that it would probably take a few transmission events for it to kind of spark and sustain transmission within a population. I think that can explain perhaps why we see in reports, you know, these evidence of early cases in a number of areas, but transmission didn't really take off until later, because with this variation, it may well be that there were exported cases much earlier, but they just didn't happen to take off in the same way. But once you saw these events where 5, 10, 20 people got infected, it enabled these outbreaks to take off. So this is the kind of paradox that once they take off, it's more explosive, but it does mean it's less likely to spark initially. The final thing I want to talk about briefly is the implications for kind of where we go next. And there's a lot of focus on these targeted measures for things like isolation and contact tracing. And I think a really nice study from a few years ago, which illustrates the challenge really of kind of what we see with COVID is this paper, which looked not only at the reproduction number, but also along the x-axis, the proportion of infections that are occurring either before people get ill or when people don't have symptoms. So you can see you've got things like SARS and smallpox, where most transmission occurs from people with clear symptoms. And that means that contact tracing can be more effective, contact tracing based on symptomatic people, because someone shows up ill, you trace their contacts, you quarantine their contacts, those chains of transmission are unlikely to go further. Something like HIV, where a lot of the transmission happens before people get visibly ill, is much harder to control through this kind of method. And I've tweaked the figure just for sort of slightly more plausible values of flu and COVID. So apologies to the authors, but if we overlay kind of based on a reasonable evidence for pandemic flu and for COVID, we probably see something in this kind of range. So flu, again, is something where a lot of transmission is happening with no or mild symptoms. Very, very difficult to control with these kind of targeted measures. COVID, slightly more feasible, but again, very difficult because you have this pre-symptomatic transmission. So by the time someone shows up with symptoms, they've already infected people. Those people are very soon going to become infectious and infected others. So you've got this very small window to work with. And that's in part why a number of countries have introduced other measures alongside. And some early work we did on this made this point that with likely features of coronavirus, that if it's taking long to isolate people who become ill, if you're missing contacts, and if you're getting, you know, a lot of this transmission occurring before symptoms that when the study was done wasn't clear, but now there's a lot of nice evidence that this is the case, you really are against the clock on these kinds of measures. And it is likely that if virus is circulating, you're going to need additional measures really alongside to be confident you can reduce transmission. And that's what we've seen even in countries like Korea that have done extensive testing. There have been additional distancing measures in place. But again, you know, having these kinds of analysis, it's useful to look more at the kind of different aspects of behavior that might be driving this. So this is a study we did a couple of years ago with colleagues at the University of Cambridge, working with the BBC, collecting large-scale social mixing data. And our hope was to make these data sets useful for a future pandemic, which occurred sooner perhaps than we would have thought. But now we are putting these into models, exploring contact tracing. So at the top, you can see different distributions of contacts made at home, made at work, made in other settings. And then the bottom, some more recent work using some of the high resolution network data we looked at to try and look at the implications of if you trace people, if you trace their contacts, how many people you're quarantining. And what you essentially end up seeing is that if you contact trace very quickly, and then if you trace the contacts of those contacts, you can effectively end up locking down small elements of the communities, because you get these kind of clusters of people who know each other or workplaces, and it almost becomes equivalent just to putting a 2-week lockdown on those individuals. But I think as well as sort of talking about transmission and R going forward, I think we also need to think about other values. You know, I've talked about R throughout this this talk, but it's not just R that depends where we go next. If we have increasing infection, if R is above 1, if control measures go in, R is below 1, the level of infection also influences where we're going to end up. You know, so if we have R equal to 1, we have a flat outbreak. But if you have level transmission when you've got a high level of infection, you're going to stay at that high level over time. If transmission can get to lower levels, ideally in a way that isn't hugely disruptive, and you can keep it at lower levels, then an R equal to 1 puts you in very different territory. So there's been a lot of focus on, is R equal to 1, is equal to 1.1, 0.9 or whatever. But really we also need to think of, yeah, if you've got an R equal to 1, you're getting 10 cases a day. That's very different to an R of 1 when you've got 1,000 cases per day. So I think as the number of infections gets lower, there's going to be other metrics I think we need to think about and really and try and get a better handle on. And one that I think is a really useful one is how many cases do we know about? So if you've got this transmission chain and you get cases appearing who are ill, how many of them are already known to your control system, ideally for your contact tracing system or your quarantine? So this is data at the bottom from Iceland that showed that towards the sort of tail end of that outbreak, a lot of the people who were detected and tested positive were already in quarantine. So those individuals were showing up as cases, but they were already exactly where they needed to be in quarantine, not in a position where they could transmit to others. So I think as we get to lower levels of infection and more targeted measures, metrics like this are going to be really important to evaluate how well we're doing, because R is a very useful measure, particularly when there's a lot of transmission. But you know, as you get more targeted, you really want to be using metrics that reflect that. So finally, I'd like to thank really everyone who's been working on this. As an outbreak, you know, we've worked on a lot over the years, but I think there's been a fantastic international effort, huge exchange of ideas globally, and particularly really impressive sharing of data, insights, models, and a lot of the work I presented is obviously work from other groups, and is really built on the incredibly generous, rapid sharing that others have done of their work. So there's a link here for more of our work there, but certainly some of the references I've flagged, I definitely encourage people to have more of a look at for more details. So thank you.
Thanks very much, Adam. That was great. That was really useful discussion about lots of the issues surrounding our estimation, and it was very clear. So thank you very much for that. We've had a few questions come in via Slido during the talk, so I'm just going to run a few through of those, if that's okay with you. So I've separated them into a few different groups. So the first one came in and it said, is R a useful measure for making policy decisions? And I note that since that question came in, you talked about a few other measures at the end, so things like the level of infection. but are there other measures that could also be useful? So for example, things like the growth rate, and how do you decide precisely which measures to use in order to make policy decisions?
I think ours a useful measure. It's not the only useful measure. I mean, as I described, growth rate, you can, to some extent, derive R from and vice versa. I think one of the values of using R, particularly in modeling, is it gives you a very clear threshold. Because, you know, if we're talking about contact patterns, if we're talking about how many people you infect, then R is a very natural way of thinking about transmission. And contact tracing, you know, you can say, right, if I've infected 3 other people and I contact trace 2 of them successfully, that gives you an R of 1. So it's, I think, a very clean threshold for understanding that sort of thing. But obviously, as we get to lower case numbers, I mean, we're seeing now there's a lot of debate around, is there increased testing? You know, what's the local reproduction number? If you get some countries that go from 2 cases to 6 cases, does that mean the R is 3? So I think we need to be looking at this kind of consolidation of measures. And particularly, as I said, you know, looking at things like, yeah, how many infections have you got, and how many can you link together? Because, yeah, 20 cases that are all in quarantine is very different to 20 random cases that have appeared and you've no idea where they're from.
Yeah, absolutely, absolutely, great. So another question that came in is to do with calculating R when you have asymptomatics and also individuals with symptoms. And the question is, can we be confident that the infectiousness of someone who is asymptomatic is similar to someone who has symptoms?
No, would be the short answer. I think that's a really good question. It's one that, unfortunately, there's still a lot of uncertainty in the evidence. I mean, there's a number of studies through contact tracing, which obviously showed people can be infected without symptoms. But the challenge is a lot of the studies are done essentially through contact tracing or this sort of thing. A symptomatic case is what triggers your study or triggers your data. And so I think we do need more data sets that basically just follow a load of people and work out who got infected, who's asymptomatic, and just get sufficient numbers, because unfortunately that is a massive unknown at the moment. And, you know, something like contact tracing, if most people are symptomatic when they're sort of transmitting or they're pre-symptomatic, that's very different to having, you know, these chains of infection where people never show symptoms.
Yeah, absolutely. Yeah. Okay, so another one's just come into that is, what is known about the precise kind of delay between infections going up and then estimated values of R increasing if you use some of the methods you described earlier for estimating R, either looking back or looking forward?
Yeah, so some of those are reasonably well established. I think the incubation period around 5 days. A few data sets have pointed towards a delay probably of around 3 weeks, maybe just slightly less, between someone becoming ill and death, if that's the outcome. I think that one of the challenging values to get at is reporting delay, because particularly during the peak of the epidemic, you know, some countries were testing people in the community quite rapidly after symptom onset, and some countries were only testing them when they got into hospital. So potentially delay from becoming ill to showing up in the data might be a couple of days, it might be a couple of weeks. And that makes a big difference, particularly if you're trying to interpret, you know, we put a control measure in on this day and what did it do? You're getting quite a lot of noise in your data. I mean, I think ultimately the closer you can get to actually when the transmission events happen, the better. But I think there's a lot of variability between countries, and probably even, you know, based on the age of cases and this sort of thing as well. Yeah,
that makes sense. So that's kind of related to that then. So if you're using R to understand the effects of interventions, can the reproduction number be used, and changes in the reproduction number be used to understand the effects of individual interventions that make up, for example, a lockdown?
Yeah, so there's been a few studies on this, and there's 2 basic ways you can go about it. So one is you can kind of constrain the model to have changes when those interventions go in. And the other is you use the methods kind of as I've outlined, where you try and estimate the reproduction number, and then maybe overlay the interventions and see if there's any obvious things that line up. I think that the enormous challenge we have, and certainly in the UK, is a lot of the major interventions went in over a very short period of time. So picking up that signal confidently in data is quite difficult. And either you need to be quite strongly constraining the model to have those changes in transmission when you think the event's happened, or being clever about how you pool information between countries, or having that flexibility, but then accepting you've got quite a lot of uncertainty. So I suspect aspects of which things were most important when are really going to be debated for years, if not decades, because of just the uncertainty of the information at the time.
Yeah, I'm sure that's true. So do you think then, if you compare between different countries, do you think that is a useful comparison to make, or do you think that actually the effects are just going to be so different between different countries that actually, you know, it all gets so blurred that there's not a huge amount you can say from that? Or do you think it is useful?
I think yeah, we need to make the best of the information we have. I mean, obviously we've got quite limited sort of scrappy bits of data often, and we need to try and say something with them. There have been a few, so yeah, that try and pool information across different interventions in different places. I mean, let's be honest, that's the big question of how do we control this as effectively and least disruptively as possible. So I think those studies are really going to be crucial, but I think just based on all the ones that have been done, you know, depending on do you use cases, do you use deaths, you know, there's always going to be some uncertainty around that.
Yeah, that makes sense. We had quite a few questions coming in about the contact matrices and estimating R when you've got different age groups. So one of them said that, so based on the DOTS acronym that you put up, you can imagine estimating different R values for individuals of different ages, because it's based on things like the probability of transmission between two different age groups. So you can imagine a kind of single R corresponding to the sort of reproduction number between those two age groups. Is that then sort of condensed somehow into a single R for the population, was one of the questions. And then a question that was related, because it was also about the contact matrices, was someone asked why the contact matrices aren't matrices that are symmetrical.
Both great questions. I'll try and give concise answers, but the first one is, yeah, completely right, that the R is a kind of this averaged value, but in reality you'd have, you know, an adult to adult reproduction number and a child to child one, and effectively to condense that down, we can use, yeah, because R is essentially this sort of linear step from one generation to the next. We can use that logic, but just with multiple populations, and mathematically you end up essentially just calculating the eigenvalue of that matrix, but it's essentially the kind of the average level of transmission averaged across the multiple populations over time, that gives you the kind of it becomes a bit abstract, but that gives you a kind of sensible averaging for that value. The point about asymmetry in matrices is in part because of population size. I mean, there's two reasons. So partly in population size, that if your age groups aren't all the same size, you might be more likely to report age group contacts in one age group than a single person in that group is to report one in your group. And there may also just be reporting biases. You know, younger groups may not report contacts of certain ages with the same probability as other groups, but often when they're converted into reproduction numbers, there's sort of steps in the framework that make sure that if there are those kind of imbalances, that they're kind of adjusted accordingly.
Yeah, that makes complete sense. So we're just gonna, because of time, we're just gonna go for one final question. And the final question says, can you tell us a little bit more about super spreading, and in particular this idea that super spreading leads to a lower epidemic probability? So would you mind kind of explaining that again? And then also, does that assume that the index case that's coming in, so the imported case, let's say, is that randomly chosen out of all the individuals in the population? And could it be the case that super spreaders, that the imported cases are more likely to be super spreaders because, for example, they travel more?
Yeah, so I think that is a good question. So in terms of the sort of probability of outbreaks taking off, it comes from this idea that if the majority of your cases aren't infecting anyone or only infecting maybe one case, then if you get a randomly sampled individual, the chances of them generating a lot of transmission, or the people they infect generate a lot of transmission, is quite low. So actually of all of your kind of introductions, very few will actually lead to transmission, but when they do, you get a large outbreak. The point about travelers is a good one, that it is making, I mean, it's making a lot of kind of quite average level assumptions. I mean, the other kind of way that feeds into is when we're talking about travel models I mentioned earlier of exported cases, you know, it's making the assumption that travelers are equivalent risk of people locally. So I think that's definitely something to sort of bear in mind in those kind of analysis. And, you know, and particularly then you get into the sort of network science bit of, are highly connected people going to interact with other highly connected people? And I mean, as I mentioned, that's why getting better data on kind of social behavior is going to be really important.
Yeah, that makes complete sense. I'm just going to go for one more actually, because we've had a couple of final ones come in. So I promise this is the last one. So someone's asked, so are there any ways to validate time varying reproduction number estimates, or to validate epidemic model predictions more generally after the fact? So if a few weeks down the line, can you say those reproduction number estimates or those predictions were kind of good ones?
Yeah, so I think there's a few ways of doing it. I mean, one is against multiple data sets. So for example, that early comics data that suggested a reproduction number dropped to, I think it's about 0.6, 0.7. Later on, when the epidemic did slow down and start declining, we started getting those values out for other regions. So that's kind of nice that multiple data sets are giving you consistent estimates. And similarly, you can calculate, say, using case data and using data on deaths. And then as data like on antibodies come in, you can get an estimate of how much the population was infected, or based on those suggested to be infected during that period, and then see if that aligns with what your model would have predicted in terms of the level of growth during that region of time.
Makes sense. Thanks very much, Adam. Thanks for a perfect plenary talk. I'd just like to also say thanks very much to the Isaac Newton Institute for organising not only this talk, but the entire programme. And thanks obviously to everyone for watching and for attending. Thanks very much.
Thank you.